Hello, could somebody please confirm that you're hearing me?
Yes.
Great, thank you. All right, if you didn't notice, I did some grading over the weekend. And I graded, I think it's lab 5. I'll look that up in a minute. And the lecture worksheet. And then I graded quiz 1A. So take a look at those, okay? Any question about any of that? Oh, there it is. Looking for my syllabus. I'm having trouble finding it. I make sure that that was lab 5. You're not seeing the screen here because I'm looking in the grade book. So worksheet lecture 4B was graded and lab 5. It was my goal to get labs 0 and 1 regraded, but I did not succeed in getting that done. And if you didn't notice, it does take me a long time to grade the quiz. If you think about it, for each question, it's going to take me at least a half a minute, probably a minute to get it graded. And there's at least 10 questions on each quiz. And there's about 20 students. So if it's 1 minute times 10 times 20, that means, what is that? 20 times, something like 200 minutes at least to grade a quiz. Okay, so be used to the fact that when I'm grading it, it's going to take me some time and I won't get it graded all the way. I like to be consistent in my answers and in my grading. So I will grade usually I grade questions 1 and 2 together on the first student, then move to the second student, grade questions 1 and 2, and then move all the way down the students. And then I began questions 3 and 4. Okay, so you will notice that it takes me some time to get the quizzes graded. And some people were getting confused about that because, I don't know, they saw only certain points and they were wondering why there weren't more points out of the quiz. It's because it wasn't fully graded yet. Any questions about any of that? Anyways, take a look at the grading. I think I got the some of the extra credit assignments graded too. I forgot about that. I'm trying to get out of the grade book and then I'll show you my screen. There we go. Oh, no, don't want to do that. Hit the wrong button there. Almost ended the meeting. All right, there, you should be able to see my screen now. So today is October 18th. And we are supposed to be starting and finishing chapter 4 today. And then we have a lab this week. I'll think about what we're going to do here. We'll probably have the lab today rather than Thursday. Any questions about what we're doing? All right, if not, let's begin. I should make sure I'm recording this. I think I am. Oh, there's the button. Okay. That didn't work. All right, last time we talked about osmosis and the passive transport of water into a cell. Passive processes are okay when the concentration of a substance that is required by a cell is higher outside the cell than inside the cell. But what about when a cell is in an environment with a low nutrient concentration, like a cell in the ocean? It's going to want glucose. Well, generally the cell is going to have much more glucose inside of it than you'll find glucose in the ocean. And then what about a cell in the desert? It's going to find very low nutrient concentrations outside the cell. So what a cell needs in environments like these, as well as other environments, it needs an active transport mechanism for moving molecules against a concentration gradient. This requires the cell to expend energy. The cell needs to perform work to move the molecule across its concentration gradient, meaning you're moving a molecule from areas where it's low in concentration to areas where it's high in concentration. Cells use active transport to pump solute molecules against their concentration gradient. This requires a transport protein, which will be a protein in the cell membrane. The transport protein is usually molecule specific, meaning one transport protein can transport one specific molecule like glucose into the cell. And you would have another transport protein to move another molecule like the amino acid lysine into the cell. This does require energy to transport the molecule into the cell. And this energy comes by utilizing ATP, meaning ATP will give the transport protein the energy it needs. And then ATP is used up, meaning it's broken down into ADP and phosphate. We'll talk about that later. Any question about any of that? All right. Active transport is very good in moving molecules across the membrane against their concentration gradient. And they're molecule specific. They happen in all cells, active transport. There is a specific active transport that's only found in prokaryotes. It's called group translocation. It does require a transport protein to pump the molecule across the membrane against its concentration gradient. It does require a high energy phosphate compound, such as PEP, and that's similar to requiring ATP by the active transport protein. And then the molecule that's moved across is transported by group translocation. But as this molecule is transported through the transport protein, the special transport protein, the molecule is chemically altered. And usually PEP transfers phosphate to the molecule, like when we're moving glucose into the cell using group translocation. When the glucose is moved into the cell, it's chemically altered and a phosphate molecule is added to the glucose. So it's no longer glucose, it's glucose 6-phosphate. This alteration prevents the escape of the molecule from the cell and allows the cell to accumulate substances more easily. Let's take, for example, a eukaryotic cell, and in the rare case, it doesn't have any glucose inside of it, and there's 10 glucose molecules outside of it. The eukaryote cell can move the glucose into the cell by active transport, because group translocation isn't around in eukaryotes. And so it moves the first glucose into the cell, and the second glucose into the cell, and then the 9th glucose into the cell. Now the 10th glucose molecule is really moving against its concentration gradient. There will be now no glucose outside the cell once that last glucose is moved inside the cell, and there will be 10 glucose molecules inside the cell. So that is really a high concentration gradient requiring a lot of energy to move the molecule across against its concentration gradient. In prokaryotes, they can reduce the cost of that molecule moving across the membrane. And that is 5 of the molecules can move by active transport. And then 5 of the molecules can move by group translocation. And so the last molecule moving by active transport, it's not 1 against 10. It will be 1 against 5 or 1 against 4, whatever. Okay. You see how group translocation is an advantage for prokaryote cells. It allows the cells to accumulate substances more easily when it's used with active transport. Any question about any of that? I'm not hearing anything, so I'm going to move on. All right. Remember, group translocation is only found in prokaryote cells. The cytoplasm is the substance inside the cell membrane. It is mostly water. It does have dissolved solute molecules in it. And that could be protein molecules dissolved in it. Even DNA, most of the time, is dissolved within the cytoplasm. Only when the DNA is highly wound up in mitosis, and then we see the chromosome, would it not be dissolved in the solution. In eukaryotes, it wouldn't be in the cytoplasm, it would be in the nucleoplasm. The nucleoid, ribosomes, and inclusions are found within the cytoplasm, but we call them a specific area of the cytoplasm, meaning there's no distinction between the nucleoid region and the cytoplasm because there's no membrane separating them in a prokaryotic cell. And then the ribosome and the inclusions have no membrane separating them. What the ribosome and the inclusions are is large aggregates or a large enough molecule that it does not go into solution in the cytoplasm. And a ribosome is just a really big cell structure, and it doesn't go into solution. You can see the dots in the actual electron microscopic image here. Any question about any of that? Somebody's microphone is on, so I'm going to see if I can turn that off. Somebody noticed it themselves and they turned it on. The nucleoid region or the nuclear area is wherever the DNA is located within the cytoplasm. There is no nuclear membrane, so wherever the DNA moves to, that would be the nucleoid region. The nucleoid region is with some stains, it does stain differently. Okay, like right here. You can see the nucleoid region, but there is no distinction. It's wherever the DNA floats in the cytoplasm. You should realize that the DNA is mostly the chromosome, but in prokaryote cells, there can be plasmids, which are a separate molecule of DNA. Plasmids are similar to chromosomes, except they're much, much smaller. Ribosomes are just the area where proteins are made. They're sort of like the factory for making proteins in the cell. They are ribosomal RNA and ribosomal protein gets together to form the ribosomes. They are so large they do not go into solution in the cytoplasm. You should know that prokaryote ribosomes are 70S in size. I think we've already talked about this. Is that correct? The S is just the sedimentation coefficient, where the ribosome sediments in the ultracentrifuge gradient. Prokaryote ribosomes are smaller than eukaryote ribosomes. So prokaryote ribosomes are 70S in size. Eukaryote ribosomes are 80S in size. You don't need to know that ribosomes have different subunits. You don't need to know what the size of those are. Let's talk a little bit about inclusions. Inclusions are reserve deposits within the cytoplasm. And the cell has accumulated so much of this reserve deposit that the accumulation precipitates out of solution, forming the inclusion. The inclusion can be used as a resource when resources are low in the cell. There are different types of inclusions. That's all you need to know. You don't need to know the names of the different types of inclusions. Here we're looking at some inclusions. All they are is molecules that there's so much in one area, it precipitates out of solution. This cell is probably metabolizing sulfur, and so it's accumulating sulfur in the cell. And then we get sulfur inclusions called sulfur granules. You don't need to know sulfur granules. There's metachromatic granules shown here. The cell is highlighted in yellow. And so there's 3 metachromatic granules. And then these are magnetosomes, which actually allows the cell to orient to the magnetic pole. All right, any question about inclusions? If not, let's move on to endospores. Endospores are a specialized resting cell that is produced by certain species, mainly in the families Bacillus and Clostridium. When the environment starts deteriorating, such that the parent cell will eventually die, the parent cell makes an endospore. The endospore is highly resistant to almost everything you can think of, such as desiccation, heat, chemicals, radiation. The purpose of the endospore is to survive conditions when the parent vegetative cell will die. An endospore is not a reproductive structure because one parent cell makes one endospore and then the parent cell goes and dies. The endospore continues. The endospore is a survival structure, not a reproductive structure. And then the endospore simply waits for the environment to change. And when the environment becomes favorable for the species to grow and reproduce, the endospore will go through germination and return to the vegetative state, meaning the vegetative state is a cell without an endospore. It will be metabolically active. It will grow and reproduce. The endospore is a cell in stasis. It isn't true that it has no metabolism, but there's almost no metabolism occurring in an endospore, meaning there's very little metabolism going on in an endospore. Sporulation is the formation of an endospore within a parent cell. And then germination is when the endospore germinates and becomes a parent cell, like the regular vegetative cell. Any question about any of that? Let me see if I can blow this up. All right. When an endospore occurs, it occurs only because the parent cell notices that the environment is starting to degrade. And so the endospore begins to be formed. How an endospore forms is the cell goes through normal DNA replication. So two molecules are made, one's a copy of the original. And then the DNA molecule moves towards opposite ends of the cell. That's all normal. We haven't talked about that yet, but that is normal. Where's my mouse dying? Okay. And then the cell starts to divide in two. With an endospore, the cell does not divide evenly like it does in normal prokaryotic cell division. The endospore will be smaller region of the cell than the rest of the cell, which we're going to call that the parent cell. And then the spore septum begins by starting to form a cell membrane in between. And then that'll separate the cytoplasm into two sections. There will then be another membrane that forms around the endospore. So it has two membranes, not just one cell membrane, but actually two. And then in between the membrane, we'll get peptidoglycan put down like a cell wall. It's not exactly a peptidoglycan because the peptidoglycan is not exactly a cell wall because it's in between two membranes. And then the spore coat forms. That's putting down specific molecules within the coat of the endospore. And then the parent cell will begin to die. And when the parent cell is dead, that will release the endospore. And now we have a free endospore. Let's go back. We got a little video if I can get it to work. Showing you endospore formation very quickly. So there's the parent cell forming its endospore. And that's it. Any question about any of that? All right. If there's no questions, that's it for the prokaryotic cells. I will talk briefly about some of the eukaryotic cell structures. Here we're seeing eukaryotic cell structures on the right of an animal cell and on the left of a plant cell. Now, you should realize that this isn't perfect, like flagellum is shown on an animal cell, but in humans, there's only one human cell, which has a flagellum, and that's actually the sperm cell. There are some plant cells that do have a flagellum. Let me see if I can remember. One is the reproductive cells of a fern has a flagellum. And then it has the it calls this a vacuole. That should be central vacuole because this is too big to be a vacuole. And it's showing it for the plant cell, which is correct. The central vacuole is only found in plant cells. There is no central vacuole in an animal cell. However, animal cells do have a vacuole. A vacuole is a small membrane-bound organelle about the size of a lysosome or a perioxysome. And these two are very similar to a vacuole. And we'll talk a little bit about that in a little earlier. It doesn't have a vesicle here, but you could find a vesicle in both the plant cell and in the animal cell. And it would be similar to in size to a lysosome. A vacuole in an animal cell as well as a plant cell is about the same size as a lysosome. Anything else? Oh, the chloroplast is an organelle you only find in green plant cells. If the plant cell is not green, it doesn't have chloroplast. Actually, that's not exactly true. But if it's non-photosynthetic, it will not have a chloroplast. Okay? And that includes root cells. They're not green, they're not photosynthetic, they do not have chloroplast. Animal cells, of course, do not have a cell wall. Plant cells do. Here's an organelle, if I can find it. Animal cells have the centrosome, which includes the centriole and the paracentriolar material. Those are used in making the mitotic spindle during mitosis and meiosis. And plant cells have a mitotic spindle, but they do not have a centrosome. All right, any question about any of that? Note that most of the organelles are similar in both the plant cell and in the animal cell. Like you can find a nucleus in both, the mitochondria in both, the Golgi complex in both. This is an actual electron microscopic image of an animal cell and of a plant cell. Do I have that reverse? No, that's a plant-like cell. It's an algae and then an animal cell. All right, this graph is showing you the main, let's shrink that, differences between a prokaryote and a eukaryote cell. There's a difference in the size of the cell. Eukaryotic cells are generally 10 times or more larger than a prokaryotic cell. There's no nucleus in a prokaryotic cell. There is a nucleus in a eukaryotic cell. Membrane-bound organelles are absent in prokaryotic cells. They're present in eukaryotic cells. The flagella are present in both. The glycocalyx can be present in both. Obviously, not all cells have a glycocalyx. Cell wall, usually present in a prokaryotic cell. There are some exceptions. Chemically, the cell wall is complex in a prokaryotic cell. In a eukaryotic cell, if the cell wall is present, like in a plant cell, chemically it's simple. Like in plant cells, it's made up of a carbohydrate. What is that called? Drawing a blank here. Sorry. It'll come to me. I'm drawing a blank. Animal cells, of course, do not have a cell wall. Plasma membrane is generally similar between the prokaryotic cell, especially the bacteria, and the eukaryotic cell. There is a difference, and that is you do have sterols in at least animal cells in the plasma membrane. There are also carbohydrates in the eukaryotic plasma membrane. The cytoplasm, this author is close to 20 years out of date when he says there's no cytoskeleton in a prokaryotic cell. That's not correct. It's a simple cytoskeleton in a prokaryotic cell. In the eukaryotic cell, it is a complex cytoskeleton. And then there's cytoplasmic streaming in the eukaryotic cell. I'll try and show you a video of that. Already talked about the ribosomes differ. They're smaller in the prokaryotic cell than in the eukaryotic cell. The chromosomes differ, at least in bacteria. They're usually a single circular chromosome. It lacks histones in bacteria. And in eukaryotic cells, there's usually multiple chromosomes that are linear. They're not circular, meaning on the X chromosome. There's a place where the X begins and a place where it ends, a linear chromosome. Oh, and the DNA is associated with histones in a eukaryotic cell. Cell division also differs between prokaryote and eukaryotic cells. Prokaryotic cells generally divide by binary fission. We'll talk about that. That's where the cell literally will divide into two. My mouse is done. There it is. Eukaryotic cells divide by mitosis. And then sexual recombination generally is none in prokaryotic cells, and eukaryotic cells, they engage in sexual recombination using meiosis. Any question about any of that? All right. We mentioned that prokaryotic cells, if they swim because they have a flagella. This isn't true of eukaryotes. They can swim and move if they have a flagella, a cilia, or a pseudopods. Of course, here's the flagella, a whip-like structure. Cilia are shorter than flagella, and there's usually many of them on the cell. They beat in unison, and so the cilia will beat that way, and that'll propel the cell that way. Okay. The third way that eukaryotic cells is not shown here. They move by flagella, cilia, and pseudopods. Pseudopods is where they put out their cytoplasm. And then they move where they're putting their cytoplasm. We already talked about that. Oh, there it is there. The cell wall of plant cells is composed of cellulose. And that's all there is in the cell wall. So chemically, the cell walls of eukaryotes is very simple compared to the cell wall of prokaryotic cells. I'm not going to test you on this, but you might want to recall in the back of your mind that fungi have a different cell wall than cellulose. And then yeast cells also have a different cell wall than plant cells. It's not cellulose in yeast and in fungi. Oop, went too far. Oh, right. Animal cells do not have a cell wall. The cell membrane of eukaryotes is very similar to the cell membrane of bacteria. There are some differences, though. They both have a phospholipid bilayer. They both have transmembrane proteins, or proteins in the membrane or spanning the membrane. They both display selective permeability, and transport across the membrane can happen by simple diffusion, facilitated diffusion, osmosis, and by active transport. Remember, there is no group translocation in eukaryotes. However, there's no group translocation, but molecules can move across the membrane by another mechanism in eukaryotes. And that's when the eukaryote uses vesicular transport, where vesicles can either bring the molecule to the membrane and then release the molecules outside of the cell, or they can bring in molecules by bringing them into the cell inside a vesicle. And that we call vesicular transport. You should know that there are two types of vesicular transport. There's endocytosis, where material moves into the cell via a vesicle. And then there's exocytosis, where material moves out of the cell via a vesicle. We usually call this a secretory vesicle because it secretes the cell's contents outside the cell. And usually it will be specific molecules the cell makes to secrete those specific molecules. And only those molecules are released, besides water. You should know there's two types of endocytosis. There's phagocytosis, where pseudopods extend around and engulf the particle. You can call that cellular eating, where pseudopods come around. I don't know if you can see this. Here's our molecule, and here's the cell, and it puts out pseudopods, which will eventually engulf what the cell wants to bring into the cell. That's one way you can get vesicular transport. And then that molecule or thing outside the cell is brought into the cell by a vesicle. There's also pinocytosis, where the membrane folds inward, bringing in the molecules, which usually bind to the receptor on the plasma membrane. And then that will fold in. Let's see if I can draw that. Wrong one. Why am I getting my ruler here? Okay, there we go. So here's the cell. Here are some molecules outside the cell. The cell will put out pseudopods. And these pseudopods will grow and grow. Until the pseudopod will eventually grow everything. And let me show you what happens there. When the pseudopods touch each other. So here we have the molecules. When they touch each other, extending from the cell, the membrane will pinch off, and then this will be the cell membrane out here. I don't know if I can write that. Not very well. That's supposed to be cell and membrane. I'm going to leave it there because I'm not doing a very good job. And then it'll pinch off over here, forming a vesicle. So this will be a vesicle, bringing the molecules inside the cell. Any question about pseudopods? Let's go ahead and show a video on cytoplasmic streaming, and we'll talk about that in a bit. I lost the wide screen. Cytoplasmic streaming is when the cytoplasm is moving around in the cell. And here in this cell, we can see cytoplasmic streaming. The chloroplasts are moving, and they'll just move around in the cell. Any questions about cytoplasmic streaming? Here you're looking at the cytoskeleton to, well, at least 2 cells, and there are 3 protein filaments you can find in the cytoskeleton. There's microfilaments, which I think is this one here. And here we're seeing most of the cell. The cell would be about right here. And the microfilaments are running throughout the cell, forming the backbone, meaning the backbone supports us. The cytoskeleton is supporting the cell. Here is the cell here, and you can actually see actin filaments, which are intermediate filaments, and microtubules in green. And there's enough of them in the cell that we can actually see the entire cell right here. And the blue region is the nucleus. Any question about any of that? The point is the cytoskeleton runs throughout the cell and gives structure to the cell. And it might give the shape to the cell, particularly for an animal cell, because the cell wall is the primary determinant of the shape of the cell, in a plant cell at least. We talked about ribosomes. Let me mention that eukaryotes do have 80S ribosomes, but there are some ribosomes that are eukaryotes that are 70S in size. In the chloroplast and the mitochondria, we do not have 80S ribosomes. They are 70S ribosomes in size. All right, so when I'm talking about organelles, I mean a membrane-bound organelle that allows the eukaryotic cell to be compartmentalized, and each compartment, meaning each organelle, typically performs one specific function. And here are some different organelles of the different cells. Let me briefly talk about the endoplasmic reticulum, the Golgi complex, and vesicles. The endoplasmic reticulum, there's 2 types. There's the smooth ER and the rough ER. The smooth ER is the endoplasmic reticulum that lacks having attached ribosomes, so it looks smooth. The rough ER has ribosomes attached to it, and then when we look at that under a microscope or electron microscope, it looks rough because of the attached ribosomes. There are some differences between the smooth ER and the rough ER. The rough ER tends to make proteins, and the smooth ER tends to make lipids. The Golgi complex is a structure that looks like a stack of pancakes right there. And what the Golgi complex does is it takes in proteins, sorts them, modifies them, and then packages them for export out of the Golgi complex. Where do the proteins initially come to that come to the Golgi complex to be modified, sorted, and packaged? The proteins initially come from the rough ER. Okay? And we'll talk about how they get to the Golgi complex and how they leave the Golgi complex. Lysosomes are special vesicles that store digestive enzymes, and lysosomes help digest things that are engulfed by the cell. Like if a cell were to engulf a particle of food, or a bacteria that's causing infection, the cell will dump a lysosome on it to digest that bacteria that has now been brought inside the cell. And that is actually how white blood cells tend to kill things that they consume. The central vacuole is a very large vacuole in plants. It acts as sort of the refrigerator for plants, meaning it stores food. It's actually the largest organelle in a plant cell. It's larger than the nucleus. It does provide support. And besides having molecules that the cell can use for nutrients like food and glucose, it also will store its waste in the central vacuole. So think of it as your refrigerator where you're putting your food, and then you're using your toilet waste and storing it in the refrigerator. That's how plant cells do things, storing both waste and then food in the central vacuole. Any question about any of that? Mitochondria and chloroplasts are unique in that these organelles do specific things for the cell, and then they contain DNA molecules as well as 70S ribosomes. We'll talk a little bit more about that. Mitochondria are where a eukaryotic cell makes the majority of ATP. So this is where cellular respiration occurs. But chloroplasts are where plant cells and plant-like eukaryote cells, meaning algae cells, engage in photosynthesis. I'm not going to talk about perioxysome, centrosomes I already talked about. Let's talk a little bit about the Golgi complex, the endoplasmic reticulum, and then the secretory vesicles, which is a vesicle, which is an organelle. And what a vesicle does is it moves material from one part of the cell to another part of the cell. Okay, so the endoplasmic reticulum, especially the rough endoplasmic reticulum, makes proteins. And those proteins are put into a secretory vesicle, which is then moved or transported to the Golgi complex. And the vesicle fuses with one end of the Golgi complex. So the vesicle comes from rough ER, brings the protein to the Golgi complex. The Golgi complex sorts the protein, characterizes them, and modifies them. I mean, it could change the protein, like add carbohydrates or add methyl groups, things like that, modify the protein. Once that's done, the protein is then sorted and then put into a vesicle where the protein then moves to where it's supposed to go in the cell. This could be outside the cell, in which case the secretory vesicle will move to the cell membrane and fuse with it. Let me see if I can show you that. So here's our cell, there's the cell membrane. I'll try and make that a little bigger. And then we have a secretory vesicle, which comes off of one end of the Golgi complex. And that's the opposite end where the proteins are brought from the endoplasmic reticulum. The secretory vesicle moves up to the cell membrane. And let me see if I can blow that up here, the cell membrane, as you recall, is two bilayers, and the vesicle will have two phospholipid layers as well. This comes up to here. Let me get an eraser now. And this part will form. So it'll fuse with the cell membrane bilayer. Have I drawn that right? Yes, I have. This will be hard to draw. I should have given more room there. Let me make a little more room. Ah. There we go. And you can see what is once inside the cell, inside the vesicle, we'll make a molecule here. This molecule is now free to come out of the cell. And that's why we call it a secretory vesicle, because it secretes stuff inside the cell, outside the cell. Inside the cell to outside the cell. Think of this as a teenager secreting hormones or something like that. Any question about any of that? So that means that the inside the cell, shrink that a little, we have an endomembrane system, at least in eukaryotic cells, where things start from the proteins, start from the rough ER, and they're moved by a vesicle to the Golgi complex, where they're modified, and then they're moved once again by a vesicle to where they belong in the cell. Okay? And that could be to exporting the molecule outside the cell right here. And then let's talk about the last thing. And that is, what happens if the protein is to stay within the cell? Where does it go? Well, the vesicle moves to where it's supposed to go. And let's say the cell doesn't want to use it right away. So it just stores the molecule inside the vesicle. Well, once the vesicle stops moving and simply starts storing things, we don't call it a vesicle anymore. So a vesicle is a vesicle that's moving the molecules inside it around in the cell. Once it stops moving and starts storing the molecules, we call it a vacuole. So a vesicle can turn into a vacuole. And in animal cells, a vacuole, all vacuoles were at one time a vesicle when they were moving. Once they start storing and not moving, we call it a vacuole. Any question about any of that? All right, I'll let you read about lysosomes. I already talked about the vacuoles. And let me briefly talk about the mitochondria. The mitochondria and the chloroplast both have two membranes, an inner membrane. Let me see if I can blow this up. Which is highly folded in the mitochondria, and then an outer membrane. Also, there's a DNA molecule, which isn't shown here, but it'll be inside the inner matrix. of the inner membrane. There's a DNA molecule within the mitochondria and in the chloroplast. There are also 70S ribosomes inside the mitochondria and inside the chloroplast. In eukaryotic cells, these are the only positions you find 70S ribosomes. All other eukaryotic ribosomes are 80S in size. Any question about any of that? So here's the chloroplast, and there's the outer membrane of the chloroplast. The inner membrane, let me see if I can blow that up, is very highly folded, even more so than the mitochondria, and it forms the granum. So inside the granum is inside the inner matrix of the chloroplast. We don't call it that, but that's what it is. Why do the chloroplasts and the mitochondria have two membranes? Why do they have 70S ribosomes? And why do they have a DNA molecule within them? Where did they come from? The mitochondria and the chloroplast. Well, the endosymbiotic theory is a theory to explain the evolution of mitochondria and chloroplast in eukaryote cells. And you can read about that. I am going to discuss that briefly. Let's see if I can bring up paint again. Oh, there it was there, actually. A long time ago, before there were eukaryotic cells, there was a proto-eukaryotic cell, and it was very big. And it engulfed and surrounded a smaller bacteria. And so that's now been brought inside the cell by, we'll say, phagocytosis or some other engulfment. Let me erase that from outside the cell now. And for whatever reason, the proto-eukaryote did not immediately digest the bacteria that it engulfed. And let me say that it engulfed that in a vesicle and then brought the bacteria into the cell inside a, well, a phagosome or a vesicle. And it didn't digest that initially. And perhaps because life was simple back then, things moved around from one cell to another. There was glucose in the proto-eukaryote cell, and some of that glucose moved into the bacteria. Now the proto-eukaryote, let me make a big text box here. The proto-eukaryote could only perform glycolysis. Hopefully I spelled that right. Meaning it could make one molecule of glucose and convert it into two molecules of ATP. And this is the proto-eukaryote. I guess I need to put that in there. I'm going to abbreviate that. PE for proto-eukaryote. It's going to take me too long to type it. Now the bacteria could engage in aerobic respiration. So it took the molecule of glucose and it didn't make 2 ATP. I got to put the 2 up there, sorry. It made up to 38 ATP. And then the bacteria released some of the ATP into the cytoplasm of the protoeukaryote. It probably wasn't 38 molecules, but it was much more than 2 ATP. And this greatly confused the protoeukaryote because it said, wow, I lost 1 molecule of glucose. Instead of getting 2 ATP, I got a number of ATP. I'm going to keep this bacteria around and it's going to make me ATP because it can make a lot more ATP than I can make from 1 molecule of glucose. Any question about any of that? So the two started living in symbiosis. The bacteria started living within the eukaryotic cell, and then the eukaryotic cell lived in symbiosis with the bacteria. The protoeukaryote gave the bacteria a home and glucose, and then the bacteria gave the protoeukaryote ATP. The cells have now lived in symbiosis for so long, we only find this bacteria inside a protoeukaryote or eukaryotic cell. We don't find it free living anymore. This is the theory. Let me go on. I'm trying to figure out where to go next. So the two have been around for so long. The bacteria is only found in the eukaryote cells now. And we don't have protoeukaryotes that now lack a bacteria. They almost all have a bacteria inside them, which makes ATP for them. And that is how they think the mitochondria evolved. Okay. And what evidence is there for the fact that the mitochondria did evolve this way? Well, the mitochondria does have 2 membranes. It has the membrane that happened or initially formed when the cell was brought, the bacteria was brought into the cell by a vesicle, and then it has the cell membrane of the bacteria. Okay. The mitochondria does have DNA, which it would if it was a bacteria that's brought into the cell. And this mitochondria does have ATP, not ATP, but ribosomes. Let's look at some other evidence. The embiotic symbiosis theory explains where mitochondria evolved from, as well as chloroplasts. For animal cells, this event happened one time, but in plants, first they got the mitochondria, and then they engulfed another bacteria that engaged in photosynthesis. And that bacteria lived in symbiosis with the plant cell and became the chloroplast. There's strong evidence for this theory. First, we can see primitive protists, which will engulf another cell and then use that cell, in this case, to engage in photosynthesis for the protist. So the protist gives the bacteria carbohydrates like sugar and CO2 and water. And then the bacterium engages in photosynthesis and then makes glucose for the protists. And then we have some, what do they call those? Other simple protists. I'm losing my thought of what you call those. Anyways, they lack a mitochondria. And what they do is they engulf a bacteria and then live in symbiosis with that bacteria. And the bacteria act like a mitochondria in the sense that I almost have the name of what this is called. Amoeba. It will give the bacteria food like glucose and a home, and then the bacteria makes ATP for the amoeba. So we actually see protists which are doing this today, is my point. Okay. And then stronger evidence is, well, first, I mentioned that the mitochondria and the chloroplast have two membranes, an inner and outer membrane. They have DNA. They have ribosomes, which are 70S in size. When we look at the growth of the chloroplast and the mitochondria, they grow by binary fission, the same way that, the mitochondria and chloroplast grow by binary fission, the same way prokaryotic cells grow. Eukaryotic cells do not grow by binary fission, they grow by mitosis. When we look at the ribosomes of the mitochondria and the chloroplast are 70S in size, the same as the prokaryote bacteria, and the eukaryotes have 80S ribosomes. Furthermore, when we look at the first amino acids made in protein synthesis, for a eukaryote, that's methionine. For a prokaryotic cell, it's a similar amino acid, but a different amino acid, 4-methionine. And for the mitochondria and the chloroplast, it is also 4-methionine. When we look at histones in the DNA, eukaryotic cells have histones. Bacteria do not have histones. The mitochondria and the chloroplast do not have histones. When we look at the DNA, eukaryotic cells have multiple linear DNA molecules. Prokaryotic cells, they generally have one circular DNA molecule. When we look at the DNA of mitochondria and chloroplast, there's one circular molecule. Furthermore, when we sequence the DNA found in the mitochondria and chloroplast, which domain do you think they fall into? Domain eukaryote, domain archaea, or domain bacteria? Come on, you got 1 in 3 chance. Hello? Anybody gonna guess? All right. The DNA sequence of the mitochondria and the chloroplast is the same as the domain bacteria. So the mitochondria and the chloroplast, by the sequence of their DNA, belong to the domain bacteria. Strong evidence for the endosymbiotic theory. Any question about any of that? All right, if there's no questions, I'm done with the lesson. I will see you at 6:30 for the lab. we're going to be talking about lab 6, I think. Yes, lab 6, disc diffusion and antibiotics. All right, bye.